we had started our Prelims Masterclass. As far as our science and technology portion is concerned, we have completed our 2 lectures. And today, this third lecture, in this lecture, we are going to continue the theme called biotechnology. In the last lecture, we started with it, and today we are going to complete biotechnology theme completely. Okay, now before we move ahead, a notification about the admissions in Narayana's Academy. Our 1 year course and 2 year prelims cum mains integrated course is already active. You can take the admissions. All the details regarding the course will be provided to you. You can go for the link provided in the description box. Just click on it and all the details will be available with you. Okay. Now, as I said, we are continuing the theme called biotechnology. Now, an important current affairs article that I found was related to biosimilars and biologics. Now, this is a very simple article, but there is a possibility wherein UPSC may ask a question on these terminologies biosimilar, what is biologic, the basic part, right? So, we need to know about it. Now, there is a news that Dr. Reddy's biosimilar of Roche's cancer drug, this one received a positive recommendation. But you will not understand the news unless we understand the basic part. Now, see, whatever medicine we get from the pharmacy, you look at the medicine. So, medicine is of two types. One is called as small molecule drug, and other one is called as biologic drug. Now, when you are making a medicine using some chemical reactions, now it's very simple. You take whatever chemicals needed, whatever chemical composition needed, in that ratio, you just bring in those elements and then your drug is formed. Such a medicine is called as small molecule drug. The best example could be your paracetamol or aspirin. These are small molecule drugs, right? Now, this is one type of medicine that we get. Another kind of medicine would be biologic drug. Now, here we do not go for any chemical reaction. We don't take any chemical components as such. Here we take biological elements, or rather we take the living cells itself. So, last time I talked about one thing, that in our pancreas we produce what we call as insulin, right? And when this insulin production does not take place in our body, or our body does not accept that insulin, then what leads us towards is the issue or disease called diabetes. Now just look at this angle. When our pancreas are not producing insulin, what we try to do is we try to bring in genetic engineering or biotechnology. We try to bring in some genes from other animals. Let's say we talk about any bacteria producing the insulin, or sometimes transplantation from a pig or any other animal, that might also happen. Now, my point is, whenever you're making use of some living cells, not chemical compositions, but living cells, then your drug becomes biologic drug, or the same term over here, that is biologics. This biologics is this biologic drug. So your insulin, which we are producing, is a biologic drug. Or let's say you're producing any vaccine. How do you make vaccine? You make use of the virus, you make use of the living cells many a times, and then you make the vaccine. So these are the two major types, or these are the categories of medicines. Now please understand one thing. When you're making this small molecule drug, making this drug is very simple, because you have to just take the chemicals that are required, and then with the chemical reaction you will get the drug. But in case you are trying to make a biologic drug, because living cells are involved, and living cells are not that simple, they don't have the same property always. You see, one human being is different from the other human being many a times, and that's the reason this part becomes very much complex. Okay, so making biologic drugs is a very complex procedure, and that's the reason it many a times becomes very costly. Okay, and one more thing, in case of these biologic drugs, in case you have made small change in the composition, or there is very minor change, that can also cause a big problem, or people might suffer because of this biologic drug if there is any minor difference also. Now this doesn't happen in case of small molecule drug. Okay. Now, why I have discussed these two? Because I had to understand what this biologics means. Okay, so we understand this is a kind of medicine over here. We understand this part. Now, you must have heard about something called generic drug. Okay, generic drug. So what we do is, let's say I have made a medicine. I have a company, pharmaceutical company. I have made a drug, like let's say a kind of paracetamol I made. Now, for the first time I have made this, so I will file patent. So I might get the patent for, let's say, next 20 years. So for 20 years, I will be making this product. Nobody can copy this process and make the same product. But after 20 years, once this patent expires, many companies will come up, they will copy my formula, copy the process, and make another drug. And here that drug is called as generic drug. So what they'll do is they'll just check the chemical composition of my drug, whatever chemicals are involved. So they'll bring in all those chemicals, make a copy of this small molecular drug. So please go back to this small molecule drug. If I just copy the chemical formula, I can make a generic drug, and this generic drug will be very cheap, very cheap. So if you go for this drug, this might be very costly, but here it will be very cheap. So we can copy and make a similar version, and this is very similar. Okay, very similar, very effective. Now in case of small molecule drug, we made generic drug. But in case of biologic drug, or in case of this biologics, when we try to make similar copy, exact similar copy, then we use this term called biosimilar. Biosimilar. Now why we are calling it biosimilar is because we are trying to make something which is similar. It is not 100% identical. It's not 100% the same biologic drug, but it is more or less similar. That's why the name biosimilar is there. And the same process will be followed. Once the patent is over, it is expired, then this biosimilar can be made by companies. And obviously, it will be way cheaper than the original biologic drug. Okay. So this is the core idea of biosimilar. Now I hope you understand what is biologics and what is biosimilar. Clear? Now see the news. Dr. Reddy's biosimilar of Roche's cancer drug has received a positive recommendation. Now see, there is a cancer drug called bevacizumab. You can read in a way you want to, but this cancer drug we already had. So it's a biologic drug, we can say, or biologic. Now this Dr. Reddy's biosimilar of this drug. So they have made identical copy, similar copy, which is called biosimilar. And this biosimilar has received some positive recommendation from European Medicine Agency's Committee for Medicinal Products for Human Use. So simply we can understand from European side, we have got some positive recommendation that this biosimilar is very good, you can use it for cancer. Clear? Biosimilar and biologics, you should be clear about it. The same stuff has been written over here. In your notes also you will find all the text. Okay, and as I told you, if you want to read all those notes, all the compilation at one place, you can go for the Narana's application. You can download it. In the free material section you will get those compilations, also these PPTs. Okay, so we're done with one topic. Please ask if you have any doubt. So if I check the comment box, no doubt. Okay. Now, the second article is related to antimicrobial resistance. Now, this is one of the most important issue at this moment when it comes to the health segment. Okay, and it's not about we Indians are facing this problem, but entire world is facing this problem. And sometimes people call antimicrobial resistance could be a new pandemic altogether for the entire world. Okay, now you need to know what is this exactly and why it is in news. So recently, global leaders They have come up with a political declaration. We call it as AMR declaration. Okay, this AMR declaration has come into picture from United Nations General Assembly, 79th United Nations General Assembly high level meeting. Now we should know what is this AMR first to understand this declaration altogether. Now see this previous year question asked in the year 2019. You can understand the relevance of this topic. UPSC has been asking on this microbial resistance part. You see, which of the following are the reasons for the occurrence of multi-drug resistance in microbial pathogens in India, right? So we can relate this question with the same theme that we are studying right now. Okay. Now, first of all, antimicrobial resistance. So for that, first understand microbes. So any microorganism that we talk about, all these are your microbes, or we use the term microbes instead of microorganisms. So your virus, fungi, bacteria, these parasites, all these come under the category of microbes, right? Now when these microbes attack our body, let's say a virus has entered your body, a bacteria is attacking your body, because of which you get many diseases, right? For example, you get TB, tuberculosis, or you get typhoid, right? Because some kind of bacteria has entered your body. So there is a bacteria called salmonella which enters your body and causes some problems, because of which you get typhoid, right? So this bacteria is a microbe. Now when these microbes, they are attacking our body, what we do is we go to doctor and take some medicines, and we call them antibiotics. We call those medicines as antibiotics. So these are antibiotics. They are going to kill the biotics. They are going to kill these microbes. So this medicine is supposed to kill these microbes. Okay. Now when you're taking these antibiotics and you make some mistakes, sometimes people overuse it. They take antibiotics more than required, or they're doing something wrong, because of which this microbe which was present in their body, that is this bacteria or virus, it becomes resistant to that drug, resistant to that medicine. So this time you took the medicine, now the bacteria has changed itself in such a way that next time if you give the same drug, same medicine, the bacteria will say, I'm not going to die now. You can give the drug, but I'm not going to die, because bacteria has become resistant. It has become stronger. It has changed itself. Okay. Now this is the stage which we call as antimicrobial resistance. That you are giving a drug which is antimicrobial, which can kill a microbe, but still the microbe is having some resistance. So you can imagine this situation wherein doctors have got medicines. Even if they give you medicines, you will not cure from the disease, because those microbes know how to deal with this medicine. They'll not die. So even if medicine is given, you are going to suffer from that disease. Now this is very dangerous. Can you imagine a situation wherein if all the populations, if they are given the medicine, still they are suffering from that disease, then what's the use of all these drugs? All these drugs will become useless. And that's why this antimicrobial resistance we consider as a situation which is very much horrific. Okay. Now, what could be the reasons? What are the causes for this antimicrobial resistance? Why we are getting this? First reason could be natural genetic process. As I said, the bacteria itself or this microbe itself has changed itself. It has become resistant. Now see, it's the part of evolution wherein all the species, they evolve with time. Whatever problems they are facing from the environment, they try to change themselves or adapt to to this environmental change. So here this resistance is developed at the genetic level, at the level of genes in those microbes. Now this is very natural thing. Second reason is overuse of antibiotics. Now this is a major cause. We're not concerned about the natural cause, we are more concerned about the human cause. What we do is we take antibiotics a lot, and most of the time I've seen people taking antibiotics when they have got viral disease. Now see, if a virus has entered your body, you should take antiviral and not antibiotic. Antibiotic is used for killing the bacteria. Okay, what you use over here is antiviral drug, which is supposed to be used to kill this virus. People are taking antibiotics. Now this will cause a problem. Okay, then many a times we see in agriculture or those livestock which are present, they are given antibiotics. Now these antibiotics are given so that their growth, they will grow faster or they'll not suffer from any disease. But because the overdose is given, people are using it without consulting any doctor, and that's the reason when you eat the meat or any material, any, let's say, milk or any product coming from this livestock, this antibiotic element is also entering your body. So this part is there. Then incomplete treatment. Many a times what we do, we go to doctor, doctor prescribes us medicine for, let's say, 5 days. We take the medicine for 2 to 3 days and then we feel that we are cured, and we don't take those medicines which are remaining for next 2 days. This is a wrong thing. Why? Because when you do this, when you've taken medicine for 2 3 days, it's not like all the bacteria in your body have died. There are few which are still there, but you feel that you are cured, and that's why you stop taking medicines. Now this, at this moment, this bacteria will have enough atmosphere present to become resistant to this drug, okay, to become more powerful over here. Then we have very limited regulation in India as well as in many countries. Nobody is checking. You can directly go to a pharmacy and get an antibiotic. It's very easy. And you see, this can become a pandemic because people are traveling from one country to the other. So one person is having this problem can be spread to other countries very easily. That's the reason we are concerned about this AMR, or antimicrobial resistance. Clear? Now there are many initiatives launched by the international community, or we can say the role of WHO becomes very important when it comes to dealing with this antimicrobial resistance. So first of all, we have this global action plan on AMR. So this was launched in the year 2015 by World Health Assembly. Now here an idea came into picture which we call as One Health approach. So One Health approach says that the human health and the animal health and the environment health, all three are linked together. Human, animal and environment, the health of each is dependent on the other. So One Health says that we have to focus on the health of all these three elements in an integrated manner. So this was your global action plan on AMR. Then came this quadripartite. So first we had WHO, then FAO, Food and Agriculture Organization, then UNEP and World Organization for Animal Health. These four organizations came together saying that we are going to deal with this AMR issue. Then we have this World AMR Awareness Week, 18th November to 24th November. It is leading as a campaign. Then Aware Initiative by WHO. You should remember this Aware Initiative. Who launched it? World Health Organization. Again related to AMR. And Global Antimicrobial Resistance and Use Surveillance System, that is GLASS. In short, GLASS, which was also launched by WHO in the year 2015. Okay. Now this GLASS is supposed to provide some information. If there are any gaps, those gaps will be filled by this GLASS initiative. Okay, these are initiatives at the world level. Now I said in the news that recently, if you go back, global leaders endorsed a political declaration on AMR. So AMR declaration has come into picture in the 79th UNGA high level meeting. So if we see this declaration, if we see the key highlights of this AMR declaration, so what all has been mentioned? First of all, it aims to reduce global deaths associated with bacterial AMR by 10%. So people who are dying because of AMR, which is related to bacteria only, they're not talking about virus or any other microbe, just bacteria. So such deaths should be reduced by 10% by 2030. This is one target. Second, national level financing. So the plan is, if you want to deal with it, you need funding, and this funding should come at the national level, at domestic level. So countries are supposed to come up with financing. Let's say you take financing or money from corporates, you take it from governments, but somehow you have to do this national financing of somewhere around $100 million, and you have to come up with an AMR action plan. This plan will be funded by this financing. Okay. Now, at least 60% of the countries should have funded national AMR action plans by 2030. Now, this is an expectation at the world level, that at least 60% countries should start their AMR action plan and funding should be provided. Then antibiotic usage. 70% of the antibiotic use, whatever antibiotics are used at the entire international level, 70% of that should be used for WHO access group. That means any segment or any drug which has been recommended by WHO. So WHO categorizes drugs. WHO provides the complete list wherein it says that in these categories, in this amount, you can provide this drug. Right? So on the WHO recommendations, 70% antibiotics should be used. Okay. Then agriculture part. So here they are saying that by 2030 we are trying to reduce the use of antimicrobials or these antibiotics in agriculture, and they expect global coordination. So I talked about this quadripartite here, this WHO, FAO, UNEP and WOAH. So this quadripartite, it is established in this meeting or in this declaration. Okay. So you should have a brief idea about this AMR declaration, highly important for this exam. Okay. Now these are global initiatives. This one is also global initiative. Now if I talk about initiatives taken by Government of India, then first thing is, in many medicines or many drugs, if you have observed, there is a red line. If there is a red line, it is expected that you should not take this drug without prescription of doctor. Don't take this drug, it's risky. So this is one thing, red line is provided. Second, National Program on AMR Containment. Now this was brought in this 12th 5-year plan, which was in the year 2012 to 2017. So this plan was brought in. So the idea was, we are going to go for the surveillance of this antimicrobial resistance. So we'll come up with the exact data, and then we can take whatever action is needed. Then we brought this National Action Plan on AMR in the year 2017. So that One Health approach which I talked, the same One Health approach is being used over here in this particular plan. And then we have Delhi Declaration on AMR. So declaration, simply ministries have come up, and all the ministries, relevant ministries, they are saying that we will do something about it. So this declaration shows a kind of commitment. Then there are state level plans also. Madhya Pradesh government came up with a plan, Delhi government came up with a plan, Kerala government came up with a plan. Even if I talk about the Kerala government specifically, they have this Operation Amrit. Now this is very famous. Operation Amrit. What people do, or what the government agencies will do, they will go for some surprise checks in the medical stores to check how much antibiotics they are selling, whether they are selling with prescription or without prescription. Everything will be checked through surprise checks using this Operation Amrit. Okay. And then we have this AMR surveillance network by ICMR. So it's a network, national level surveillance and research on AMR is going on through this. So all these are initiatives. You should have a brief idea about these initiatives. So overall, we have completed this antimicrobial resistance topic. Okay. Now, any doubt till now? Yes? No doubt? Fine. We're done with two topics. Now the next one. Now this one is called as naphthromycin. Now the name itself, you can understand, the name seems like a kind of drug. So it's a drug which has been recently launched by government of India, and its name, actually in the market, the name is Micnaf. What is it exactly? It's an antibiotic drug. Okay. Now we need to know what is the speciality of this drug. Okay. So come here. Key features of nafithromycin. Now the speciality is, right now we are facing the problem of antimicrobial resistance in many people, in many patients. So now we are bringing some drugs which can deal with this problem of antimicrobial resistance too. So we brought this medicine. We are saying this can deal with a problem. Let's say a person has got pneumonia. Okay. Earlier we were giving other medicine. Let's say we were giving this azithromycin as a medicine to that patient, just for the sake of example. Now the patient has become antimicrobial resistant towards this medicine. This medicine is not working on this person. Now we have got another medicine called this nafithromycin. We are saying it can be used for typical bacteria or atypical drug resistant bacteria. Means normal bacteria which has entered for that, instead of using this drug, you can use this one drug. Or in case the patient has become, or the bacteria in that patient has become resistant to that drug, in that case you can use nafithromycin. So if a person is suffering from this problem of AMR, in that case this medicine can be used over here. So if we have got this one medicine, maybe in future we'll get many more medicines which will be made for atypical drug resistant bacteria. Okay. Then few more features, like it is more effective, it is 10 times more effective than this azithromycins. And usually what happens, doctor gives us 6 7 10 medicines, you don't want to take so many. Now you have a good news, because this nephromycin, it requires only 3 days treatment regime. So only 3 days medicines will be provided, and that's why doctors are saying the compliance from patient side would be on a higher end, because patients are okay with taking medicines for 2 3 days, not more than that. Okay. So such is the medicine called nafithromycin. Now this is India's first indigenous macrolide antibiotic. That's why it is in news. And it is developed by Wokhart, and the Wokhart got some support from Biotechnology Industry Research Assistance Council, that is BIRAC or BIRA. So in the last 30 years they have researched on this, and with the research they have come up with this particular medicine. So just try to remember the name nafithromycin for prelims. Clear? Easy? Very easy, right? Now, next topic is also very important. There can be a question which is application based. Many times UPSC asks application based questions from science and technology portion. So this topic, environmental biotechnology. Okay, you might expect some application based question over here. Now see, environmental biotechnology. We know what is biotechnology. What is environmental? That means when we are using biotechnology to solve some of the problems which are related to environment. So the problems could be your climate change, the problem could be your global warming, or problem could be anything related to environment. When you are solving those problems, you are coming up with a product or something which can solve those problems. There we are dealing with this subject called environmental biotechnology. Okay. Now the recent news was that there are many advancements taking place in environmental biotechnology. So we are thinking about a potential solution in this particular field when we talk about climate change, because climate change is concerning for us. So maybe we will have solution in this field. Now, when we talk about this environmental biotechnology, first you can see the diversity in terms of biotechnology. When we talk about biotechnology in terms of environment, wherever we are using it. So there are many categories. There are total 10 categories which we see. So we use this term called red biotechnology. Red biotechnology, when we say this thing, we are talking about the healthcare part. So you are making vaccines, you are making insulin or whatever. There you are using biotechnology, and we call it red biotechnology. One thing. Second is your green biotechnology. Now this is about agriculture. So to improve the crops, or let's say crops have to be made resistant towards some pests, then in that case you're talking about green biotechnology. Then third category is white biotechnology. Now this is about industrial processes. For example, you have this biofuel which you make in the industries. So this is about white biotechnology. Then yellow biotechnology. This is about food production. So in the food, when we are improving the nutritional quality of the food, let's say I talked about a rice variety, golden rice. So in that golden rice, we added vitamin A. Additionally, we improved its nutritional quality. So that part will come in yellow biotechnology. Then we have blue biotechnology. Anyone can understand this is about the marine resources. So whenever you're using marine resources, where you're trying to utilize that part, then you're dealing with blue biotechnology. Then gray biotechnology. Now whenever gray part comes, then you can think about some part which needs to be cleaned. So whenever you're trying to clean the environment, then you're dealing with gray biotechnology. So to conserve the environment, we have to clean some elements. So let's say I give you the example of oil spill that takes place in oceans many a times. So you try to make use of biotechnology to solve that problem. We use the technique called bioremediation. Okay, bioremediation, I'll discuss that part. But what you do is you take one bacteria or you take one microorganism and you send it in that ocean wherein this bacteria is going to take all that oil. So that oil will be removed from the ocean. Very good. This is bioremediation. Now this will come in this gray category. Okay. Then gold biotechnology. We're using the term gold. This is used for bioinformatics sector. Now, bioinformatics, here the biotechnology, this is being added with the information and technology part. So this is your gold biotech. Then brown biotechnology, when you talk about any desert area, arid region, there if you are trying to make use of biotechnology, let's say you're trying to make some species, some plants which are resistant, which can survive in these arid regions or through this genetic modification, then that is part of brown biotechnology. Okay. Then you have got purple biotechnology. Whenever you deal with the legal issues, ethical issues, let's say related to patents or biosafety, that is purple part. And the last, black biotechnology. The name black very clearly shows when you make use of biotechnology or these microorganisms for killing peoples, for murdering someone, or let's say for terrorism or war, then that part would be black biotechnology, manipulating microorganisms for biological warfare and bioterrorism. That is your black biotechnology. Okay. So this is where we see the diversity, and this is where this environmental biotechnology comes into picture, because we see how we are utilizing biotech in different aspects. Okay. Now I'll give you some examples, some key applications of biotechnology in environmental field. So there are a few things called as biomarkers and biosensors. Now there are some things which we can use, means here we are obviously making use of some biological process or some microorganisms, some plant species, something we are using, and we are calling them biomarkers. Now these biomarkers are some kind of indicators. Let's say you have got a pond over here, or you have got something over here, and in that there are some pollutants, there are some toxins, it is highly contaminated. But how will you know whether it is contaminated or not? Its health you can understand through these biomarkers. So let's say there is some water in which heavy metals are present, you can understand with some biomarker. Similarly, there are biosensors also. These are some devices wherein we are using some biological process, let's say some enzymes, some microbes, some tissue, something we are using. They are also sensing that there is a toxin present in this water body. There is some contaminant, some pollutant is there. So these help us. Certainly these help us. And then this bioremediation, which told you just now, that oil spill takes place and we make use of a bacteria or microorganism to remove all that oil. So this is bioremediation, which is the best example of environmental biotechnology. Then you have got biofuels. Now we are heading towards renewable energy sources. We don't want to depend on fossil fuels. We are looking for different sources. When we look for different sources, at that time we look at biofuels. Now these are the fuels which are made from some biological process or some living cells or living beings. Earlier, the fuels that we were using or we are still using, like petrol, diesel, these are made from fossil fuels, right? But here we are making use of bio, that is some living part. So let's say we have different biofuels. For example, first generation includes biofuel coming from food crops. Let's say we're making use of corn, sugarcane. We're going for some process with those corn and we're coming up with some fuel. That's the first generation biofuel. Initially, we started with making biofuel using these stuffs, which are food crops. Example is bioethanol, biodiesel. So initially, we started making this. With improvement, we came up with the second generation. See, earlier we were using food crops. Later on, what we did, we started using crops, but not the food crops. For example, we started using crop residue, we started using woody plants, because you see, food crops are needed for human beings to eat also. Our food security is also important. So if you utilize all the food crops for making biofuels, what are we going to eat? That's the reason now we reach to the second stage. We are using the residual part. Anyway, we are not going to eat that, right? Then the third generation involved algae and microbes. We started using algae and microbes to make biofuels. Now, this is something very interesting. If you're using algae, first of all, this algae is very helpful for you because it is going to sequester carbon dioxide. You look at any green plant, what they do is they go for photosynthesis. When they do photosynthesis, they take carbon dioxide from the atmosphere. They go for some reaction and they make food for themselves. Now carbon dioxide is being absorbed. When you look at algae which is green is also going to absorb some carbon dioxide. So it is helping us somewhat. It is helping us remove some carbon dioxide. So this third generation, after this we reached to fourth generation of making biofuels, in which we started using genetically engineered crops. So we were making modifications at the genetic level and now we started using those crops to make our biofuels, and these are carbon negative fuels. Carbon negatives means whatever carbon is getting emitted when you are burning these fuels. Let's say this carbon is of this much amount, then the carbon it is actually absorbing will be this much. So they're absorbing more and giving less, emitting less. That's why it is carbon negative fuel. Clear? I'm saying this biofuel is one of the example of our environmental biotechnology. Okay. And the last would be this molecular ecology. This is also one good example. Now, when we talked about biotechnology, we make use of many tools. Like last lecture, we talked about this CRISPR-Cas9. It's a tool to cut the gene at a particular location. Now, when we use such tools, such genetic tools like CRISPR-Cas9, or let's say there's something called DNA fingerprinting, when we are using all this, we call it molecular ecology. Okay, DNA fingerprinting is very interesting. Just like you have this fingerprint, which is very unique for you. Similarly, if you see the DNA pattern, that is also very unique. Every person will have a different DNA pattern. So that pattern we understand using this DNA fingerprinting technology. So all this is part of this biotechnology, environmental biotechnology. Okay, this is application part. So there could be some statements given directly and should be in a position to answer it. Okay, so we're done with this topic, environmental biotechnology. Let me see the chats if we have any doubts. Okay, now the next topic. It's a very easy topic, I would say. The topic name is xenotransplantation. It was in news many a times in the newspapers. Xenotransplantation. Now see, simply understand what is xeno. Xeno means any foreign species. When you look at yourself, we call auto. When you look at other species, other organisms, we call it xeno. So we are using the term xenotransplantation. You know the transplantation. When you take any tissue from some other person and add it in your body, or when you are moving any cell or any organ, then that is transplantation. Okay. So xenotransplantation. What happens is scientists have been trying to do one thing wherein they were trying to transplant many organs, cells, tissues because we have this demand. We have this demand. If you go to hospitals, you'll observe that there are many patients who are looking for some donors who can donate any organ, any tissue because they need it, right? We get this news that kidneys needed or some other organ is needed. Now there is very high demand for such organs or tissues or something. That means transplantation is needed. But the problem is there are very less donors. There are very less human beings who are ready to donate any such thing called your organ or tissue, or even in a single cell could be part of it. So donors are less, but the demand is very high. Now here we see a gap. And scientists thought we can bridge this gap. We can bridge this gap using xenotransplantation. Xenotransplantation would mean we will try to bring in organs or tissues or cells from other animals, other species. Let's say you take a pig. Let's say my heart isn't working properly. I'll take the heart of a pig and transplant it into my body. It's not just about this one heart or something. There could be many other organs. Let's say we were making insulin, right? So insulin is produced in our pancreas. So you do one thing, you take some cells from the pig's pancreas and add it into our body, right? This is what we call as a xenotransplantation. Now for the first time this xenotransplantation in humans took place in 1980s. The first heart xenotransplantation took place in 80s. Now, one problem with this xenotransplantation is there is a possibility of rejection of organs. Let's say you are trying to add any organ of pig into my body. There is a possibility my body will not accept it, will completely reject it. My immune system will find it a foreign particle and my immune system will directly start attacking that organ. So, what's the use of this transplantation? It becomes completely useless if you do this, right? This is one challenge, and that's why scientists follow a step by step approach wherein what they first do is they go for genetic modification. So let's say they're taking something from pig, some organ or some tissue or some cell. They will go for genetic modification. First they want that this organ or this part should be accepted by my body, so they will add some genes from my body to it. They'll make some modification at the genetic level so that it is accepted by my body. Obviously for that genetic engineering part they'll use the tools like CRISPR-Cas9. Okay, this will be done when they're looking at those animals. Let's say they're selecting pig or any other animal. Even those animals should be very healthy, so that part has to be considered. Okay, then the transplantation will take place, all the surgery part wherein you're taking this organ and putting in my body or any patient's body. Transplantation will happen, and one special drug is given to this patient which we call as immunosuppressant. What happens is, even if you do all these genetic modification and everything, still there is a possibility that my immune system, my body's immune system might still attack this new item which is coming from some other's body, some other person's or some other animal's body. So what doctors will do, they will give a special drug called immunosuppressant. So what it does is it is suppressing our immunity. It is saying to our immune system that just calm down, don't attack, calm down. So such things are given after the surgery. After this surgery, these immunosuppressants are given. And after that, sorry, after that these patients are monitored. Follow-up is taken, they are monitored for a long duration. So we can say this xenotransplantation, it's a lengthy but very interesting process, and somehow we are trying to bridge this gap of organ shortage which is there. Okay, so xenotransplantation, very easy to remember. Okay, now the next one. Next one is your bio ride scheme. Bio ride scheme. Now it's a very simple scheme but very important scheme. Considering the necessity towards this biotechnology field, government has come up with a scheme which is bio ride scheme. You can read the name. Okay, bio ride. That means biotechnology research, innovation and entrepreneurship development scheme. Such a long name, in short, is called as bio ride. Now what is it exactly? Who brought this? Union cabinet has recently approved this particular scheme, and what it is exactly going to do is one thing. We have observed that our scientists who are working in the laboratories, they are coming up with different experiments, they are coming up with new research. Now they have come up with substantial research in the field of biotechnology. Okay, the research is completed, lot of things are present with them, but the problem is nobody is acting on it. Means they have done some research. Let's say for cancer, for the sake of example I'm taking, for cancer a specific drug can be made. For that, the research has been done by our scientists. But there is no company who is making this particular drug. There is no company, there is no entrepreneur who is taking up all this research knowledge and making a product out of it. Okay, so there is a gap which has been observed, especially in this biotechnology field. So to bridge this problem, bridge this gap, government has launched this bio ride scheme. Now, what are the objectives of this particular scheme? First, promote biotech innovation and entrepreneurship. So, innovation and entrepreneurship, this is a major part which will be promoted by government. Government will provide some incentives. So, this gap will be bridged, and there is a target which has been set, which is 300 billion bioeconomy by 2030. Okay, now you're looking at biotechnology not from the perspective of technology but from the perspective of economy. So how you can apply whatever technological knowledge you have got, how you can apply, make a product, you can make this biotech industry better and contribute in economy. So $300 billion is the target that we have set already. Now if we look at the details of this particular scheme, the implementation agency is your Department of Biotechnology. We have a ministry called Ministry of Science and Technology. Under this ministry, we have Department of Biotechnology, and this department is going to implement this scheme. Budget allocation 9,000 crore almost, and the timeline is for next 5 years, or this 2021-22 to 25-26. So this was the timeline already set. Already it was set, but now obviously it has been approved very recently, so we can understand that it might be extended. Then it is merging three components. One is the research and development part, which is done separately. Second is the industrial and entrepreneurship development part, and third is biomanufacturing and biofoundry. Now, what is biomanufacturing and biofoundry? This part you should know. Whenever you are producing any good, any product you are making, by making use of biotechnology, by making use of some biological system or process, then such a product or such process of producing such product is called as biomanufacturing. Okay, using biotechnology you are making a product, that is biomanufacturing. Biofoundry, now wherever you are doing this biomanufacturing, that facility, okay, it's not like normal manufacturing will be there. There will be many things like you have synthetic biology, automation, computational tools, all will be available over there. So that facility where all this thing will happen is called as biofoundry. Okay, so this is very technical stuff that is available with us, and this is biomanufacturing part. Now this is also added and all three components are being merged in this particular scheme. Okay, so they're saying that we are going to give some incentives, some grants to people, for example in the field of synthetic biology, biopharmaceuticals, bioenergy, bioplastic, whoever will be working in these fields, we will give some incentives. This is what the government is saying under this scheme called bio ride scheme. Okay, good, good to go. Now the next topic is bioplastic. Anyone has any idea about bioplastic? Any rough idea about what is this bioplastic all about? No? Plastic, we all know plastic. We're using stuffs which are made up of plastic. Now we are talking about something called bioplastic. So, if you know one thing about plastics, they are infamous for one thing which we call as they are not biodegradable. Okay, we are talking about plastics. When we talk about the normal plastic that we are using, they're non-biodegradable. They don't degrade easily, or even if they degrade, it takes somewhere around hundred and thousands of years for them to degrade. They don't decompose easily. See, normally if you've eaten, let's say at your home, you're cooking and you've thrown some waste material out of it. Now that waste material, if you put on the soil over here, on the empty soil, after some time you'll see that some decomposition taking place, that material will degrade. But if you put a plastic, that is not going to degrade. And because of this, we face this problem of plastic pollution, because it is not degrading, it is remaining over here on this planet for a very long duration. I'm saying in the next 100, 200, 300 years, the plastic that you have produced and thrown over here, it's going to stay here. It's not going to get degraded. Now that's a problematic stuff, and that's the reason nowadays what we see, the problem of plastic pollution in different ways. In oceans you see so much plastic. Even this plastic will come to us in the form of microplastic, very microplastic element. All these things are happening. We are concerned about plastic. So now we are talking about a new concept wherein we are making use of biotechnology to make a kind of plastic that we call as bioplastic. Okay, making use of biotechnology, we are making a plastic. Now what we do over here, we make exact similar kind of plastic, but this plastic is made up of vegetable fats, oils, cornstarch, straw, wood chips, food waste, etc. Now you may ask, sir, what is the normal plastic made up of? The plastic that we use normally, it is made up of fossil fuels. So when fossil fuels, we get this petroleum crude oil and all, it goes to that center wherein all these processes go on to make a product out of it. Let's say they are making petrol out of it, diesel out of it, kerosene out of it. So in the same process you'll also get the plastic. Okay, the plastic that you use or the polythene that you use, so even that material is coming from there. So fossil fuels are the source of normal plastic, but these bioplastics, they will be made from these sources: vegetable oils, vegetable fats, cornstarch, straw, wood chips, food waste, etc., etc. So the source is more or less biological and more or less plants. We are using plants to make this product. Okay, now when you look at bioplastics, few things you should know. One is this bio based bioplastic. Bio-based, that means it's a bioplastic. Simply, we are making use of some plants or something. Now, there are some which are degradable and there are some which are non-biodegradable. Okay, the same bioplastic could be non-biodegradable and it could be biodegradable. Now, this is something you should know. So here some examples are given. I suggest you to read them, revise them. There's nothing to explain about it. If I give the example of non-biodegradable bioplastic, this P, polyethylene, PET, polyamide, polytrimethylene, terephthalate, all these are examples. And there are some biodegradable ones. So PLA, polylactic acid, PHA, polyhydroxyalkanone, what is the name? Alkanoids. Tough name, but you have to remember PHA, PLA, PHB, PGA. All these are biodegradable. Okay. And this third category that we need to read is biodegradable fossil-based bioplastic. Some bioplastic will make using fossil fuels. And I'm saying these are biodegradable. Plastic, normal plastic was not biodegradable. These are biodegradable. So examples would be PB80, PBS, PBOH, PCL, PBA, etc. Okay, you can read this and I suggest just revise it once or twice at least to remember this part. This is bit technical but important. Okay, done with bioplastics. Now remains the next topic, that is artificial wombs. This is also very easy, I can say. Artificial wombs. See, now we are trying to make wombs which are artificial. Obviously, wombs are natural, present in the females, and in the reproduction process, wombs play an important role. The baby is staying in those wombs for let's say 9 months, that gestation period. Now we are saying, let's say there are some problems. Let's say there is this problem called premature premature birth which is taking place. Sometimes, see, the gestation period or the period for which the child should be there or the fetus should be there inside the mother's womb, that is approximately 9 months. But sometimes we see, because of some complications or some problems, premature childbirth takes place. So the childbirth takes place before completion of that 9-month period. Now this means the child has not developed all the organs or everything. When we say the development of this fetus, in this case, when this premature birth is taking place, this development has not completed properly. The child is not completely developed, and before the complete development of all the necessary organs and all, we see the birth. Now, this could lead to many complications. The child might face many problems after getting birth. So, to help this child or this fetus after coming out of the womb, now we are trying to develop this artificial womb. So we are trying to make artificial womb which is very similar to this natural womb. Whatever this womb provides to the child when it was inside the womb, all things will be provided to this child through this artificial womb. So what all will be provided? So there's one thing called amniotic fluid. So when the fetus or this child is inside the womb, it is usually provided with this amniotic fluid, and this fluid provides all the nutrients needed to this child, right? So now we will provide the same amniotic fluid to this fetus once it is outside, okay, through this entire system. Okay, then all the nutrients, oxygen supply, child also needs the oxygen supply. That oxygen supply will be provided. There will be one oxygenator. You can see over here, this is the oxygenator. So this oxygenator, it replicates placenta. So whatever placenta does in the mother's womb, same stuff will be done by this oxygenator. So overall, you should just remember these terms: amniotic fluid, this oxygenator, and all the nutrients and oxygen supply which is needed, that will be provided. So there's something called cannula which is going to act as umbilical cord. We have umbilical cord through which everything is moving, everything is being passed from mother to the child, whatever nutrients, oxygen supply, whatever is needed. So this cannula will be there. So this is how we have made artificial womb. Okay, so although I can say this is at the research and development part till now. Right now we have made it in theoretical terms, not in practical terms. That means in biotechnology, or when we are studying about all these terms, there is research still going on. We are still not in a position to claim that we can make artificial womb, but we are trying to make it. We are very near to making this stuff, and this is going to revolutionize human reproduction, is what this article is saying. Okay, so this is all about this artificial womb, and we've completed our biotechnology theme. So till now we have completed substantial portion of the science and technology current affairs articles. Substantial as in, I'm not saying have completed a lot, but substantial, right? Because biotechnology is a major theme, as I said. So we completed it. Now in the next lecture we'll start with the next theme. Okay, so for today we'll wrap up the session over here. If you have any doubts, you can please ask. Any doubts if you have for this lecture, please ask. I'll just wait for 1 or 2 minutes and then we'll wrap up the session. Yes, any doubt on these topics which we have discussed till now? No? Okay then. So with this we will wrap up the session. We'll meet in the next lecture tomorrow, same timing, 12 p.m., and we'll cover the next theme. We'll cover all the articles from last 1.5 years presented in the newspapers in the last 1.5 year in the next lecture. Thank you so much for joining.